And now they are ethicist Alex London.
While I'm waiting for my slides, I have to say I felt a little bit blindsided this morning. I didn't know that there was going to be a food metaphor running through the conference, so I would have brought my pierogi game from Pittsburgh, but I apologize for being late on that. So when I went through the decision aid, I had trouble with the first and the third and the fifth boxes, and I thought I would try to put together my thoughts on what my concerns were. I have to start with just something you know. Trials are tools for answering specific questions, and but from the ethics standpoint, the nature and the source of the uncertainty that the trial is designed to answer is critical to the ethical evaluation of the design. And design features have to be justified by their contribution to the quality of the evidence that they're going to produce, and by the relative risks and costs of alternative designs relative to the information that they would produce. So it's true, as the algorithm lays out, that obscuring intervention allocation can frustrate patient and provider preferences. And so I started to think, look, where are we when we go through this algorithm? Where are we in the life cycle of development? Because development isn't a linear process. It's a recursive process. As you change the questions of efficacy will return again. So has efficacy been established? Does honoring preferences confound our estimates of efficacy? And which component of the things that go up to make real-world effectiveness are we really examining? So I want to say something about real-world effectiveness. It's really a function from two variables that have a bunch of parameters, right? The first is the intervention ensemble. So this is a term Jonathan Kimmelman and I used in terms of thinking about what the structure of clinical translation is. Drugs themselves are probably just toxic substances. In order for them to produce some therapeutic effect, you have to know the dose to deliver them, the schedule, the features of the population, where I'm not convinced that we know all populations are linear, so that we know the space is, we say, oh, well, we did it in this population, and then it's sort of going to drop off. I mean, there can be pretty steep drop-offs in the population variable. What co-interventions you have to use, what diagnostic requirements. For us, those are all part of the intervention. And those are different from utilization factors, patient-provider preferences. How much does the thing cost? What's its tolerance? What's that likely to translate into in terms of adherence? Do we have the clinical capacity to deliver this intervention to get the most out of it that we would like? And is there even awareness? Clinical practice can lag behind. So are people aware of the evidence supporting this intervention and how to use it and so on? So features that can affect patient and provider preferences are things like double single dose, oral injection, medical surgical, less testing or more, better and worse tolerated. So I want to try to represent now the situation which I think we hope to be in after we carry out a bunch of our research. And I think we have to think about research as portfolios of study. And that's one of the things that goes into coloring my view of, say, the Salford study. If you view it as a single study versus if you view it as a study that's part of a larger portfolio of evidence that was generated. So our whole portfolio should put us in something like this position where we know the relative efficacy of these two interventions. We know that this intervention has, more attractive to patients. It's single dose, it's easier to use, something like that. And so we're pretty confident that will translate into superior real-world effectiveness. So when I write things in black, that means, we're relatively confident. We've got in the data that we have for that. Now you're in the situation where, well, what happens? Oops, can I go back? Yeah, sorry. So what happens when these things diverge? We say, well, intervention ensembles, one is more likely to be more efficacious, but this other one has these utilization factors that might make it more attractive. This does seem to be a case then where it's probably not a wise decision to blind, Precisely because what you're trying to ascertain is the degree to which those factors are going to be what influences real-world effectiveness, right? So does the lower efficacy but greater utilization, adherence, and so on translate into a better effect in the real world? So I think that can be a legitimate undertaking as part of a larger portfolio of studies. What happens when we're in this situation where we have evidence about relative efficacy, we have evidence about what the utilization factors are going to be like, but we don't have a lot of evidence about the inequalities here? And it seems to me that this can be a situation in which you might not want to blind, but it's going to depend a lot on what the policy question is that you're trying to ask. Do you want to identify the most efficacious intervention and then try to change practice? You might snicker, but I think part of what we're doing is we're trying to change practice all the time. That's got to be part of the variable, right? There's too much, even to make the whole underpinning of this, of the current meeting go forward, we've got to change practice. We've got to standardize data recording. We have to standardize practices and procedures. So I think a lot of what we do is we say, let's find out efficacy and then let's make that translate into the real world by making our clinical practice, realize that efficacy in practice. There are going to be other situations in which you might say, listen, let's let these things vary. Let's hold this fixed. Let's let those things vary. And then let's see what we get in terms of, the most real-world effective intervention. I can imagine situations like that. I guess for me, the question is, I need to see where we are in terms of what the whole portfolio of evidence that we've generated for this product looks like. So I write the first ensemble in red now to sort of illustrate that if we're not really sure about efficacy, then I think the deck starts to get stacked against blinding or obscuring the intervention allocation. When expectations are based on efficacy without sound evidence, then they're just likely to be false. The philosopher Hegel said that history is the slaughter bench of human happiness. And on Twitter, I've said clinical trials tend to be the slaughter bench of clinical certainty. There's a lot of stakeholders also who need to rely on the evidence that we're generating from trials. It's not just the clinician treating the patient, but the trial evidence goes back into drug development generation, what happens when we're treating patients who are refractory, and so on. Without sound evidence, I'm reluctant to rely on real-world effectiveness instead of ascertainment of efficacy. Sham surgeries were mentioned, and I think they're a lovely example. The sham knee surgery for osteoarthritis of the knee, that was a procedure that was preferred. You had clinicians and patients who sought it out to the tune of $650,000 a year and about $3.25 billion of expense. The trial was a single-blind, randomized sham surgery control where you were allocated either to arthroscopic surgery or a sham arthroscopic surgery, which was largely theater. They read a script, they performed some manipulations on your leg behind a blind, they made surface cuts in the skin and then bandaged you. The sham here was essential to obscure outcome intervention allocation, which was essential here to getting a picture of the intervention's efficacy, and in this case, the actual surgery was not superior to the perception that you had had surgery. We can blind across modalities. I only put this up as an example of a recently published protocol for migraine prevention. It's real acupuncture versus placebo medicine. You're randomized to that or to sham acupuncture and topiramate. So these are two examples of shams that have some greater cost to participants associated with them. They're not the most dramatic shams that I could have produced. There are cannulas into the brain that deliver saline in Parkinson's trials. So they're extreme cases, but the methods that we use to blind or obscure outcomes can carry affirmative risks. Those need to be disclosed to participants. but they need to be offset by the value of the evidence. And so again, how much value we get for the evidence from the blind is going to depend on what else we know, what the other trials are in this portfolio. But I do want to say, when the concern is, well, will patients enroll in a trial like that? I think part of the thing is we have to not give biased informed consent. We do have to say, this is the risk that you undertake by getting this, whatever procedure we're using to mask. But these are also the risks that you take from getting the active arm if it's not effective. So you wouldn't want to have a sham surgery, but when we talk about the control, but you also don't want to have a sham surgery that's very invasive because it's the real thing, it just doesn't work. Finally, one of the things that we talk about is extending the label. I think this is a really tricky thing. I think, yes, I like the focus of this meeting. I think we also need to push to have clinical trials that have more realistic populations in them and not settle for either or. The thing about invested, right, it's got a lot of pragmatic elements to it. But in terms of focusing on efficacy in this different patient population, there I think it's important to make sure that you take away that element of uncertainty in order to be sure about efficacy, because I would say this isn't so much of a pragmatic endpoint here as more of an efficacy endpoint. So my final points are just that the nature of the uncertainty should influence the trial design. Uncertainty about relevant risks and potential benefits has to be clearly communicated. Part of that too is what kind of uncertainty do you have? There's many types. One is we just don't know. The other is there are partisans for A and there are partisans for B, and we want this trial to generate the evidence to know who's right. And we have to communicate to patients and participants the role of this design in addressing that uncertainty. Thank you.